Hi, Miss Nikki here. Welcome to Chapter 3. By the title, you can see we're going to talk about cell structure and function. Here's a list of the two fundamentally different cell types that exist. I kind of think of them as a large umbrella, right? So here's our, I'm going to abbreviate, prokaryote, and here's our eukaryote. So basically, what kind of examples would fall under prokaryotic? Most people automatically think of bacteria. They do not have a specific nucleus. So basically their DNA is just kind of free floating. Eukaryotes, some examples that we'll see coming up, this would be plants, animals, right? So these possess a nucleus. So kind of these are the two camps. You can either be prokaryote or you can be eukaryote. Before we move on to talk about prokaryotes and eukaryotes, I want to talk about cell theory. And again, I know we've talked about this word theory, but go back to Chapter 1 so we can just have a refresher. Scientific method, and we know that you have a hypothesis and you'll carry out an experiment and then you'll collect some data and you'll review it and you'll come up with a conclusion, right? If you do this multiple times, if everywhere you look and you come up with the same answer, the same conclusion, we usually start calling that a theory. So this cell theory is saying that all organisms, anything that's living, is composed of cells. So most of us go, okay, yeah, we know this. We didn't know this back in the day, right? We didn't have microscopes. We couldn't see cells, but now we see it all the time. Cells are the basic living unit of structure and function. All cells can only come from other cells. So you don't have spontaneous generation. So if you do a little history about cell theory and about this all cells come from other cells topic, they used to think if you were selling meat in a market and within 5 days there were now maggots on the meat, they thought that dead meat could spontaneously generate maggots. We know that's not true, right? We know that a fly had to land and deposit eggs that eventually turned into the larva. So you have to start with a cell to make a cell. And we haven't found any organism that doesn't have at least one cell. So this is cell theory. Along with cell theory, let's talk real quick about surface area to volume ratio. So you can see all three of these have the same volume. Do they take up the same amount of space? Yes. So they have the same volume. But in terms of surface area, the area around each individual tiny cube here is much greater than just this one large cube. So if this was a cell and it was really large, if we made a protein product here, I made a protein, and I needed to ship that protein out of the cell, it would take a really long time to get there, right? It would create a lot of energy to get it out, and it would take a while. Well, here, what if I made a protein product here in a really tiny cell? Well, I don't only have to go a short distance to be kicked out of the cell. I couldn't think there. I was going to use the word exocytosis, but we don't know what that is yet. So basically, this entire slide is basically saying, why are cells small? So cells are small, and then they're grouped together into tissue, because by having a really small cell, you have increased surface area compared to volume, and this allows us to take in more nutrients and release more waste and do it more efficiently. So let's move on to prokaryotic cells. So one, they're going to lack a nucleus. So there's no membrane-bound nucleus. You're just going to see this kind of string of DNA. It's free-floating. Two, underneath the banner or umbrella of prokaryote, we have two domains, bacteria and Archaea. I tell people just forget this A is there. It's Archaea. So again, kind of that umbrella I was drawing, here's the whole class of prokaryotes. And then we're going to have subclasses. One's going to be bacteria. The other subclass is going to be archaea. So these are similar in that they're both prokaryotes, but they're different enough to put them in separate classes. When we look at eukaryotes, I talked about it before, we're going to see plants and animals. Again, same kind of concept here. They're similar in that they both fall under eukaryote, but they are very different, right? We cannot take the sun and turn it into sugar. 3, they're usually unicellular, right? One-celled. And then this is just something to kind of pay attention to. Not all bacteria cause disease. So we usually think of bacteria automatically as bad because the only time they're in the news is if they make us sick, right? But there are some that are beneficial. So you'd be surprised probably to learn that in your small intestines you have E. coli. Some of these bacteria help us break down the food that we eat and help us extract the vitamins that we need in order to survive. Here's a nice example of a bacterial cell. So you can look through each of these, look at the little hair-like bristles, that there's kind of this stringy DNA. Sometimes they call it the nucleoid region because sometimes it squishes into like one section of the bacteria. Plasma membrane, cell wall, they have a capsule so that they can stick to things. They have a tail, flagellum, and they have ribosomes. Ribosomes are what's going to make protein. So we have DNA that's going to be transcribed into RNA, and then with the help of ribosomes, we're going to make proteins. Same thing with the bacteria. So I would look through this slide, see if it makes sense to you, and make sure you can identify some of these terms. And then we're going to move to a table that we have in your book as well that's nice. So here we have all three, and we're kind of doing a comparison. So remember, these two are prokaryotes, and then we have our eukaryote. So what's the difference, or what's the similarity between these two? Do both archaea and bacteria have a plasma membrane? Yes. Do they have a nucleus? No. Do they have organelles? And organelles are kind of subcompartments. And we'll see this when we look at eukaryotes. No. Do they have ribosomes? Yes. So just as kind of an added thought, we haven't talked about prokaryotes in great detail, but you can see some of the similarities here. So do all prokaryotes and eukaryotes have a plasma membrane? Yes. So all cells have a plasma membrane. Do they all have ribosomes? Yes. So this is all cells have ribosomes. What's the only big category of cells that has a nucleus and has organelles like mitochondria and chloroplast, right? This is going to be eukaryotes. So there'll be a few questions on the exam where if you took a sample and you put it underneath a microscope and you saw a certain structure, what would it tell you? So here I mentioned an organelle is mitochondria. This is what produces ATP, or energy for the body, right? So what if you put a slide sample underneath the microscope and you see a mitochondria? You know it has to be a eukaryote. Now we move on to eukaryotic cells. They're really complex, right? They're going to have subcompartments. That's going to be organelles. They're going to have a nucleus, and inside that nucleus is going to be DNA. And that these are the groups, the 4 major groups. I know I just mentioned plants and animals in the beginning. Those are the ones we're the most familiar with, but there are 4 major kingdoms of eukaryotic cells. This is a nice table from your book that kind of organizes everything for you. So we're looking at the structure of eukaryotic cells, and it goes through each one and talks about what it's made up of and what its function is. So we do see cell walls in some eukaryotic cells. So we have plants, fungi, and some protists have cell walls. This is something that's also similar with prokaryotes, right? Bacteria also have cell walls. So this is a structure that they share, but you can see that eukaryotes don't all have cell walls, right? Animals missing here. Animals do not have, say, no cell walls. So it's good when you're looking at these tables to get an idea of how they're similar, how eukaryotes and prokaryotes are similar, and how they're different. Plasma membrane, all. So everybody has a plasma membrane. You have to have a plasma membrane in order to be a cell. So it's all prokaryotes and all eukaryotes have a cell membrane. Nucleus, only eukaryotes. So you can see nucleolus or nucleoli, get it out, ribosomes. We said yes, all, right? Everybody's got ribosomes because everybody's got to make proteins. Rough endoplasmic reticulum and smooth endoplasmic reticulum, you're only going to find these in eukaryotes. And the table continues. Golgi apparatus, lysosomes, vacuoles, peroxisomes, mitochondria, chloroplast. This chloroplast you're only going to find in plant cells and some protistas. Cytoskeleton, cilia, flagella, they're rare in plant cells. And then centrioles, this is animal cells only. So they make a good kind of highlight here, right? So centrioles are going to help with division. So you're only going to see these in animal cells. The thing I wanted to mention is, as you go through these, some of the components that every cell has to have, and I kind of alluded to this with plasma membrane. So you're always going to have a plasma membrane. You're always going to have ribosomes. You're always going to have DNA. That does not mean a nucleus, right? Prokaryotes don't have a nucleus, but they still have DNA. So all cells have DNA, ribosomes, plasma membranes, and then some people like to also include cytoplasm, which is the jelly-like substance that's inside of the cell. So basically, it doesn't matter if you have a cell, it has to have these items. And there are rare exceptions to almost every rule. I always say in biology, never say never, because biology will make you a liar. When you look at this image, I would go through each of these and make sure that I can identify the structure name and have some idea of what it does. So here's an animal cell. Obviously on the outside, we're going to have a plasma membrane, right? Inside, we're going to have a nucleus. And they use this word chromatin. It's the same as DNA. Chromatin is just kind of stringy DNA. A nucleolus, this is what produces ribosomes. So you have to have a nucleolus to make the ribosomes, which ribosomes are going to do what? They're going to help you make proteins. The orange little jelly beans, these are the mitochondria. Let's see, centrioles here. These are microtubules that are going to help during cell division. You're going to have a Golgi apparatus right here. You're going to have rough endoplasmic reticulum. The rough is studded with ribosomes. And then you're going to have the smooth endoplasmic reticulum, and this is going to help create lipids. So I would go through this image, make sure that I can identify structure and have some idea of what's happening. Something nice to notice is here's the DNA, right? What's our flow of information? DNA to RNA, and then with the help of ribosomes, we're going to make proteins. And then we're going to ship those proteins somewhere in the body. So here's my DNA. My DNA is going to be copied into RNA. My RNA is going to be read by the ribosomes, and I'm going to put out a protein product. And then that protein is going to be put in a vesicle, and it's going to be sent to the Golgi apparatus, which is going to tag the protein and fold the protein. And then we can transport that protein within the cell or outside of the cell. So we move on to plant cell anatomy. You can see from some of these tags that some of them are very similar to animal cells, right? Smooth ER, rough ER, ribosomes, nucleus, Golgi apparatus, plasma membrane. We know everyone has to have a plasma membrane, right? So what are some of the differences? That's kind of what I would focus on with the difference between plant and animal cells. Know what they have in common and then know what's different. So chloroplast here. Chloroplast is the structure that's actually going to carry out photosynthesis. So turning sunlight into sugar happens in the chloroplast, and plant cells and some protists have chloroplast. Also, this central vacuole, this allows the plant to maintain that kind of internal pressure. If you think about a crisp piece of lettuce versus a wilted piece of lettuce, the wilted has lost water, the vacuole has gotten smaller, and that's where we have kind of drooping plants. So this ability to store water happens in the central vacuole. So central vacuole is different, chloroplast is different, and cell wall is different. So there's no cell wall in animal cells. We do see it in some bacterial cells, but the difference between plant and animals I would kind of focus on. So again, that question, if I said I've put a organism that I found on the microscope and you're looking at it and you see a chloroplast, what does it have to be? 9 times out of 10, it's going to be a plant cell. Is it possible it could be a protist? Yes, but there's very few of those. They're rare. So it's mostly plant cells. This slide is basically talking about the transport outside of the cell. I kind of showed you on the other image, but its technical name is called the endomembrane system. So here's my nucleus. I'd have DNA. I'd transcribe it into RNA. The rough endoplasmic reticulum would turn it into a protein. It would get shipped to the Golgi apparatus. It would be tagged and sorted, and then that protein could be excreted outside of the cell. My favorite topic here, energy-related organelles. So we're going to talk in chapter 8 about chloroplasts and how plants carry out photosynthesis. And then in chapter 7, we're going to talk about mitochondria and how they generate ATP energy through a process known as cellular respiration. So the plants take sun and they turn it into a carbohydrate, and they have oxygen and create oxygen, right? So they release a carbohydrate and oxygen. That's going to be picked up by the mitochondria, and it's going to be used to generate ATP energy, and CO2 is released. And we know that plants absorb CO2 and the sun initially to make this carbohydrate. So that's why they're kind of showing you this circle. Something to pay attention to is plants have both. So plants have both chloroplasts and mitochondria. So plants don't make food for us. Plants don't make carbohydrates so that we can consume them and have energy. Plants turn sunlight into carbohydrates, and then they use the carbohydrates in their own mitochondria to generate energy for the plant to grow. Here are the reactions of cellular respiration and photosynthesis written out. So photosynthesis, sun plus carbon dioxide plus water yields carbohydrates plus oxygen. In cellular respiration, we take the carbohydrate plus the oxygen and we turn it into carbon dioxide and water, and we store the energy in the bonds of ATP. So we can do this, or the plants can do this, right? So cellular respiration happens in both plants and animal cells. Now I figured we'd talk about the cytoskeleton just a little bit. We're going to look at 3 different types: actin filaments, intermediate filaments, and microtubules. All of the cytoskeleton is going to help maintain the shape of the cell. This doesn't seem like such a big deal now because every cell we talk about is basically round or kind of a more box-shaped. But in the human body, if you were to take anatomy and physiology, we have things like neurons, which have these dendrite fingers and have this long axon tail, and then have these little feet. So this neuron structure is pretty complicated. How do we maintain that structure? Well, the cytoskeleton is responsible for that. So think of cytoskeleton as kind of like the scaffolding inside of the cell. It can assist in the movement of the cell and the organelles, moving the organelles around inside the cell. And then also has this kind of dynamic feature in that some of them can assemble and disassemble. So you needed to build a bridge, you can do that, and then you can tear this bridge down. There's a really nice video that I have linked inside this chapter where you can watch the inner life of the cell on YouTube, and they've done a really good job of showing you kind of things that would likely happen inside the cell or things that we have seen, but it's more of like an animated version. But it's still really nice and worth watching. So the first group we're going to talk about are actin filaments. These can form underneath the plasma membrane and kind of provide some structure, so like scaffolding, right? They also form projections inside the intestines called microvilli. So the plasma membrane of the intestinal cells kind of goes like this, and then it forms these fingers. This has to do with that surface area. So remember, intestines, you're absorbing food, right? So you have lots of points of absorption. That happens because of this structure, and this structure, actin filaments are responsible. So you can see how everything's kind of tied together. A little more detail about these actin filaments. So remember, we're still talking about cytoskeleton, right? This actin filament helps us move, muscle movement. And real quick, when I say muscle, I'm not just talking about skeletal muscles, the ones that are attached to our bones. I'm talking about your heart. It's made up of cardiac muscle. You also have smooth muscle, which helps pump blood through your blood vessels. So actin filament plays a role along with these myosin protein molecules that actually pull or contract muscle so that you can move. Next, we have intermediate filaments, and these are in between, right? That's why they're called intermediate. So actin and microtubules. So actin would be kind of the largest, and microtubules would be the smallest, and then in between you have these intermediate. So these form support for the nuclear envelope. This is what's around the DNA and creates the nucleus. They also help form cell-to-cell junctions, so holding all of your skin cells together. Might sound silly, but you know how if you burn and you can peel skin off in a sheet, right? That's because these intermediate filaments were holding all those skin cells together in that sheet form. And then this also helps strengthen human hair. So you're going to find intermediate filaments in hair. The third type of cytoskeleton that we have, category, I guess, is microtubules, and these are going to be hollow cylinders. So they're made up of this tubulin, and they form kind of this hollow tube-like structure. We're going to look at two classes of microtubules up next because they're important for humans. We talked about centrioles earlier when we were talking about animal cells. So the reason that we talk about these is that we believe the centrioles are kind of the hub that are involved when microtubule assembly and disassembly happens during cell division. So you have these centrioles, these rod-shaped structures, and we think that microtubules start radiating, they're building, right? And they build out from these centrioles, and they help direct chromosomal movement. So we'll talk more about this in cell division, just kind of have an idea that centrioles are going to have something to do with microtubule assembly and disassembly to aid in cell division. Another type of microtubule assembly happens for cilia and flagella. So remember, flagella we saw with bacteria, but in humans, the only cells that have flagella are sperm cells. Cilia we have lining our airways, like your trachea, bronchi, and these cilia help kind of sweep any particles that make it into your trachea, try to sweep them up and out. The last topic we need to talk about is the evolution of eukaryotic cells. So again, remember when we come up with some of these theories, we're saying we've tested these over and over again. We've carried out all of these different experiments and we keep coming up with the same answer. This is our best guess to date. And I'm going to have you guys do a discussion topic on this so we can talk about it a little bit more. But the fossil records suggest that the first cells were prokaryotes. We have not found in sediment layers, the earliest sediment layers, we haven't found any eukaryotic cells. So the fossil record says the first cells were prokaryotes. 2, biochemical data suggests that Archaea are more closely related to eukaryotes. 3, eukaryotes evolved in stages from prokaryotes. So number 2 here, what they're kind of saying is we have bacteria, and then we have this Archaea, and then we have eukaryotes. Remember, these are both, bacteria and Archaea are both prokaryotes. But this Archaea seems to be kind of in between, the intermediate. So the latest theory about how mitochondria and chloroplasts, which are prokaryotic in nature, how they became part of a eukaryotic cell. And this theory is called endosymbiotic theory, or sometimes called endosymbiosis theory. So they believe that mitochondria were originally bacteria, and that chloroplasts were originally, cyanobacteria is a term for a group of bacteria that can turn sunlight into energy, carbohydrates. So both bacteria, and that somehow they entered a host cell and they begin to live cooperatively. So let's look at some of the supporting evidence for this theory. Both organelles, so I'm talking about chloroplast and mitochondria, they're similar to bacteria in size and structure. So they look very similar to other bacteria that exist outside of a eukaryotic cell. They're bound by a double membrane. So we have a cell, and that cell has a plasma membrane. Inside, if we're a plant cell and we've got chloroplast and mitochondria, they have a double layer membrane. A lot of people are like, why? Why would you have a double layered membrane? For me, this is probably more relevant that they have their own genetic material. Why would they have their own genetic material? So chloroplast has its own DNA. Mitochondria has its own DNA. This is separate. This is additional to the DNA that's in the nucleus. That's strange, right? None of our other organelles have their own DNA, just chloroplasts and mitochondria. They also divide by splitting. Splitting is something that bacteria do. They have their own ribosomes and they make some of their own proteins. So from this DNA, they're going to transcribe their own RNA. They have their own ribosomes that they make, and they make their very own proteins. So these are proteins that were not directed by nuclear DNA. RNA-based sequences suggest prokaryotic origins. And a lot of this we've started finding out more and more about as we've done more genetic studies, right? So we're going to do a discussion on this. I'm not telling you you have to believe this. I'm just saying there's a lot of food for thought here. Here's a proposed image of how this might have happened. So they show a prokaryotic cell, and that somehow the mitochondria and the chloroplast were brought into the cell, and that would account for the double membrane. If you had your mitochondria and all of a sudden it's engulfed by the plasma membrane of the original prokaryotic cell, that would account for this two-layer membrane. Having the mitochondria be separate and then brought in also explains why there's more, or why it has its own DNA extra DNA, right? So again, this is our best guess to date. When you're looking for articles to post on the discussion board and talk about this, you're going to see that there are some good reasons for this. And there's also people that say, well, this is why I don't believe that this theory exists. So just make sure that you check your source. You know, I always jokingly say Billy Bob Joe's website might not be a good place to find accurate information, right? If you go to a creationist website, they may be biased, right? Just like if you go to a science website, they might be biased. So you really have to pay attention to what your sources are and try to find alternate sources and kind of compare, read between the lines, and come up with your own theories and thoughts. So it'll be fun to see what you guys have to say in the discussion board. Okay, well, this is it for chapter 3. I'll see you next time.